Welcome to Barry's in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Well, welcome back to In the Interim. I'm Scott Barry. I'm your host for today of In the Interim. I've an interesting topic for today. I used to write a column for Chance Magazine, and I would quarterly write a column called A Statistician Reads the Sports Pages. And of course, consuming and talking about things that show up in sports, the statistical analysis of this. I did that for about 10 years, actually. In that it was very rewarding. So I'm going to do that today, and this is A Statistician Reads JAMA. So I'm going to tell you about an experience of opening JAMA and giving a read to relatively a random clinical trial that I read the results of, and it sparked a number of things I thought very much worth discussing. By the way, for those of you out there, whether you're driving to work in the morning, the afternoon, you're out for your daily run, or however you consume In the Interim, I'd love to hear from you. Let me know what kind of topics you would like to talk about, particular people you'd like on In the Interim. Love to hear what you'd like to hear about. Here at Barry Consultants, we have a company of about 35 scientists, mostly statistical scientists. We work on clinical trial design. We work on implementing adaptive trials. We have software for simulating trials. We do a wide range of therapeutic areas. And yes, we focus on innovative trial designs, adaptive trials, Bayesian statistics. And so I'd love to hear what kind of topics you'd like to hear about on In the Interim. So here we go. So this really happened. I got an email from JAMA, and in there they list different articles and results of different trials. And unfortunately, I don't get to read these as much as I would like, but one showed up and I thought, you know what, I'm just going to read one of these and see what these trials look like. So this trial is the FAIR HF2 trial, and it was published, the primary author is Anchor. So Anchor et al., and it says published online March 30th, 2025 in JAMA, in an original investigation. So if at all what I talk about is interesting, yes, please go check out the trial. I'm not involved in the trial. I had nothing to do with it, and I only vaguely know a couple of the authors, so I don't have any involvement in this trial. So what is the question in the trial? The trial, and it says this right in JAMA, what are the efficacy and safety of intravenous ferric carboxymaltose? I'm going to refer to this as intravenous iron supplement. The paper refers to it that way. So this is for in patients with heart failure and iron deficiency. So is this intravenous iron supplement for patients with heart failure and they have an iron deficiency? Is it effective and safe? That's the question in the trial. Okay. So very interesting. So the design, the settings, and the participants, it's a multi-center trial. It's randomized 1-to-1 with the iron supplement in there. Patients with heart failure, defined as having a LVEF, left ventricular ejection fraction, less than or equal to 45%, and having an iron deficiency. I'll let you, if you know what levels of iron deficiency are, I'll let you go to the paper to see that. By the way, they define a particular highly deficient group, and I'll say something about that as well. But so everybody's iron deficient, and there's a particular subgroup that is highly deficient. It enrolled at 70 clinical sites in 6 European countries. Enrolled from March 2017 to November of 23. Median follow-up of patients was 16 months in the trial. Okay, so again, any of the details, please read the article. So sounds very interesting. The trial enrolled 1,105 patients. So a rather large trial from that perspective, large and long trial. The primary endpoint in the trial, and it's a bit sort of interesting, and I'll try to lay this out a little bit. So the primary endpoint, it's looking at cardiovascular death and heart failure hospitalization, very common endpoints in heart failure trials. It's going to analyze 3 different primary endpoints, where that's an endpoint tied to an analysis in the trial. So the first one is the time to first cardiovascular death or heart failure hospitalization. Again, a very common way to analyze in heart failure trials is a time to event. So they're doing standard time to event analyses for that endpoint. Additionally, they're doing an analysis of the rate of total hospitalization. So this is a patient could have multiple heart failure hospitalizations. And they're using rather standard analysis techniques of that for count data, negative binomial analyses for count data, depending on the exposure the patients have. The third one is analyzing the first endpoint that I described, which is time to first cardiovascular death or hospitalization, but it's restricted to that subgroup that I described that are highly deficient. And so those are the 3 analyses that are going to define the primary set of analyses in the trial. Yes, they're going to control the overall family-wise experimental error rate by analyzing all 3 of those analyses. So sort of co-primary, if you will, in the sense that any one of those could potentially be successful, and they adjust for that. Okay, so they use a Hochberg procedure for analyzing those 3, I'll call them endpoints, and I struggle with this a little bit because I like to think of endpoint as heart failure, time to heart failure hospitalization or cardiovascular death. And how you analyze it or the subgroups aren't really the endpoint, but I'll describe it that way. I think it'll be easier to describe it. So they are analyzing these 3 analyses. And in the analyses, they've set up a procedure where they will refer to this as statistically significant if any one of the following 3 things happen. If all 3 of those are significant and I'm going to do it in the one-sided sense. In one-sided, this is all about superiority. They describe it in the paper 2-sided. So I'll do it 2-sided, sorry. So if all 3 are less than 0.05, 2-sided, then the trial is statistically significant and they've demonstrated superiority. The second opportunity is that if 2 of them are significant at 0.025, so that's half of the original, but if 2 of them meet 0.025, then the trial demonstrates statistical significance. If any one of them is significant at 1.67%, 2-sided, then it demonstrates statistical significance. So this is sort of 3 shots on goal where they're looking at 3 different analyses. Again, time to cardiovascular death or hospitalization, the number of heart failure hospitalizations, and a subgroup where they analyzed time to first cardiovascular death or heart failure hospitalization. Okay, so the SAP is published as part of it. It's a very well-written SAP. The design is reasonably standard. I found no evidence of any adaptations in the trial, so they enrolled 1,105 patients and carried out the primary analyses. I'm sure there was a DSMB and safety was reviewed, things like that, but a very traditional trial, and a good trial. And I'm going to talk about the publication of this and the results of this. The authors should be commended on this trial, the patients involved in the trial, and they deserve praise. So I hope in no way does this come across negative for the people who conducted, and published this trial. But I want to dive into the science of it. I want to push a little bit on the science of it, and I want to give my reading as a statistician who randomly picked up this article and read it, what my reading of it is. Okay, so the structure's set up. What happened in the trial? So the trial enrolled 1,105 patients. Again, it was randomized, double-blind in the setting. The first analysis of time to cardiovascular death or heart failure hospitalization, they report this as the number of events per 100 patient years, as just a way to summarize it in the paper. Is in the treatment group, it is 16.7, and in the placebo, it's 21.9 per 100 patient years. So 16.7 to 21.9. 141 of the patients of 558 on the treatment had an event, and 166 of 547, reasonably similar sample sizes. So 141 and 166. The hazard ratio in the time to event analysis is 0.79. The two-sided p-value is 0.04. One-sided would be 0.02 of superiority. The hazard ratio of 0.79 is showing the treatment did better. And notice that doesn't meet that endpoint by itself in the setting of the Hochberg procedure. If that would have been the lone primary analysis, it would be statistically significant. Now, what happened to the other endpoints? It met 0.05, so if all three meet 0.05, the trial will be considered significant. The total heart failure hospitalizations had 264 in the treatment group and 320 heart failure hospitalizations. Now that's adding across patients. It matters how many patients have 0, 1, 2, and 3, and so on. The relative risk in that analysis is 0.80, 0.80. Again, a benefit for the treatment. A 20% relative risk reduction in heart failure hospitalizations. The p-value, the two-sided p-value is 0.12 in that. The third, which analyzed this subgroup of patients that met this high need, showed and this was the endpoint of cardiovascular death or heart failure hospitalization, time to first event, showed a hazard ratio of 0.79. Also, exactly the same as the primary analysis, the confidence interval is a little wider and the p-value is 0.07. So let's think back to the Hochberg procedure. Do all of them meet 0.05? They don't. The first one did, the other two didn't. Do two of them meet 0.025? No. Actually, none of them meet 0.025. And none of them met 0.0167. So according to the primary analysis methodology, the controlling of the experiment-wide type 1 error rate, this trial is not significant. Statistical significance was not shown. Again, a very interesting result that time to cardiovascular death or heart failure hospitalization showed a significant p-value. So for example, the confidence interval shown goes from 0.63 to 0.99. That's the 95% confidence interval. So what is the conclusion in the trial? So I'm reading this, I'm looking at the data, I'm looking at the results. The conclusion and relevance for the paper: in patients with heart failure and iron deficiency, iron supplement did not significantly reduce the time to first heart failure hospitalization or cardiovascular death in the overall cohort, or in patients with transferrin saturation less than 20%, or reduce the total number of heart failure hospitalizations for placebo. And in the little figure they show that talks about the population, it's really very nice. It's the cartoon of the article. The conclusion says that iron supplement was well tolerated but did not significantly improve outcomes compared with placebo in patients with heart failure and iron deficiency. Okay, so what does a statistician, and by the way, this is just me as the statistician, I'm sure other statisticians have a very different reaction to me. So I read this and I really, really struggled with this from several points. And so let me sort of dive into the points. The first part about this is just the scientific struggle I have that we report trials as black and white. They're significant or they're not. And in this trial, where the only reason it's not significant is because that overall cohort on cardiovascular death and heart failure hospitalization was part of a multiple testing procedure. So it was incredibly close to being significant, but it wasn't. The conclusion of this trial is that iron supplement doesn't help patients, doesn't change the clinical outcome of patients. A Bayesian analysis, say, of that primary endpoint would say there's, assuming a non-informative prior, would be a 98% probability that iron supplement benefits on time to cardiovascular death or heart failure hospitalization. So any way you want to read this, there's gray area to this. Every trial typically has gray area to this. But yet, when we publish it, it's all or nothing in this scenario. The conclusion is the same as if the data were identical in the two groups, and the hazard ratio was 1. Even if it showed harm, the conclusion would be the same, that the treatment doesn't benefit. And really, conclusions say one thing: the treatment benefited or it didn't. I think it's a gross simplification of a 6-year trial of 1,100 patients. But I understand how we got there. And by the way, statisticians share some blame in how we got there. We reinforce that hypothesis testing and type 1 error of 5%, and you can't say anything if you don't reject the null. And that's the way we analyze trials. This trial, I bet if you flip 3 deaths in this trial, it meets the Hochberg procedure. And our conclusion to clinicians reading this article is that it benefits. It's either it doesn't benefit or it benefits. Now, we hope anybody reading this dives into the data, looks at the results, thinks about other trials, thinks about the treatment of this, and makes a decision based on it. But I have to believe the conclusion from JAMA, it makes a huge indent into any clinician reading this article. So the black and whiteness of trials, I just feel like is scientifically, I really struggle with it. And I accept that as a member of the statistical community, we're probably partly to blame for this dogmatic approach to hypothesis testing, and it's black and white. And we're going to come down hard on you if you don't interpret it any other way than that. Okay. And now for those statisticians out there, I have nothing against the Hochberg procedure. And I know that we design trials. We do FDA trials where 5% scenario, 2.5% one-sided tests is the standard. And we live by that. And we do Hochberg procedures. So I don't have anything against that. But I really struggle with the likelihood principle aspect of this. The data in this scenario is exactly the same as another scenario. The data are identical where this paper says it's statistically significant and this iron supplement benefits patients with heart failure and iron deficiency. This 0.05 had the exact same data set. And the likelihood principle that if we have exactly the same data in two different scenarios, our conclusions should be the same. If you're a Bayesian, the posterior probability is identical for those two trials. The Bayesian machinery satisfies the likelihood principle by the nature of the Bayesian Bayes theorem. So I really struggle with this part of it. And I get it from a type 1 error scenario. Being a Bayesian, I don't think type 1 error is the be-all, end-all. And it flips this result for clinicians reading it. And I really struggle with that. Okay. Now, given the rules we play by, and they knew the rules they played by, they wrote them in this article. So the SAP lays out superiority. They knew that if they ended up in this situation, so if somebody simulated this trial and showed them this result, by the way, that's a huge value of simulation. If they saw that result and said, good, that's the result we want, great. But I'd be really surprised if that's the result they wanted in this trial. There's no adaptation. Could this trial have been adaptive? Could we have seen that result? Could it have been bigger? Now, I recognize this was a 6-year trial. And it may be the funding of this, it couldn't have been bigger, and this is just the way it is. And then they would say, the investigators would say, yes, that's the result we want. But I think it's one of those scenarios that had this trial been 6 months longer, had it enrolled 200 patients, would it have changed clinical practice? Does this paper change clinical practice? Should it change clinical practice? Would it have changed clinical practice if the trial were 6 months bigger, 200 patients bigger? That's a little bit of the struggle here. And so I just bring out that I hate to say it, but is 1,100 patients in 6 years, is it wasted? In the way, because we do black and white. Now, we shouldn't do black and white, and I'll talk more about what would be shades of gray. But these are the rules that everybody plays by at this point. Journals play by this. We kind of know the rules going in. Could it have been adaptive? The other struggle I have is that this is about science. This is about recommending treatments to patients that could potentially benefit. And I would think if the truth of this is a hazard ratio of 0.8 on heart failure hospitalization and cardiovascular death, this is a clinically important treatment. So we only analyze data in the trial. We are stuck on that. By the way, I think that's reflective of frequentist approaches. We analyze the data in the trial. We calculate the probability the data is as extreme or more extreme than what we saw, assuming the null. That's the p-value, 0.04. But this is, there's science here. We typically know more. Part of my struggle is what's next? This trial, if you're into giving adjectives, it's borderline significant. It's very close to being statistically significant. Do we need another trial that's 6 years in 1,100 patients and get the p-value of that next trial below 0.05 or below Hochberg stuff? Or would 200 patients potentially, I said, suppose that trial was bigger by 200 patients or 300 patients. Would it change clinical practice? But no, we can't do that. The next trial designed would only analyze that trial. There's something incredibly frustrating about that. Now, I want to give you a different potential scenario. Suppose this was a novel treatment. I assume that nobody owns the rights to this. Nobody has patent life on a iron supplement, and this is all about treating patients. But suppose this was a novel treatment in heart failure, and a company ran this trial exactly this, and they don't get significance. And they go to a regulatory agency. They go to EMA, they go to PMDA, they go to the US FDA. And they say, we just can't approve it. It's not enough based on that trial. Does that company need to run another trial of 1,500 patients? Can we say, look, there's information there. The next trial doesn't need to be as big. We're really close to approving this, but we just can't do it. And there are examples of this. Do we start over? It seems bad science. Now, the FDA is absolutely doing this. There are scenarios where they use the results of one trial combined to the results of another trial. You can look up Rebiota. It was approved in 2023. Ferring Pharmaceuticals. There are multiple devices that have been approved this way. There's multiple scenarios that I know of that we're working with the agency or have designed trials where it uses the previous results. Recognizing that, boy, it's really close. We shouldn't need 1,500 patients after this for approval. So I want you to think about just the medical community in that scenario where we're so focused on single trials. What about combining the results together of this? My biggest issue with this, as a statistician reading it, is I don't believe the conclusions. I think they're wrong. Now, maybe it's just me, but I think this treatment works. And let me give you a little bit of why. So when I read the article and I see that, first of all, the statistics is compelling to me. That's highly likely, just based on the trial, if we're stuck on only this trial, a 98% probability for a really clinically important event is really valuable. In a scenario like this, where this isn't about an FDA approval, which has its own regulatory standards, and I know we've got to go by this, but this is about the next patient that walks in the door. If it were me, I want iron supplement. I think it works. It's highly likely to work. But the other thing I thought is, okay, we do get type 1 errors. We do get scenarios where we get a hazard ratio like this and a confidence interval, and the treatment doesn't work. So as a statistician, I want to know what other information is out there. And what do we know about this? Well, there have been previous large trials run, and the trial nicely, it's actually, you can't really find much about it. There's a little bit in the JAMA article, so I don't want to say there isn't, but it talks about the reason and still the uncertainty about this. And so there is a trial, the HEART FID trial, that it looked at time to cardiovascular death or heart failure hospitalization. All three of the trials I'm going to tell you about that have already been published use that same endpoint, time to first cardiovascular death or heart failure hospitalization. And that trial showed it had a hazard ratio of 0.93 for that endpoint, with a confidence interval that went up to 1.06. So that probably had a p-value something like one-sided 0.1, two-sided, maybe 0.15, and maybe a one-sided 0.075. I didn't go find the article, but I found the summary of that. So hazard ratio of 0.93. The Iron Man trial, a great name for the trial with an iron supplement, had a hazard ratio of 0.84 for that same endpoint, where the upper bound of the confidence interval is 1.02. Borderline significant, but not. That trial did not demonstrate clinical benefit, but hazard ratio of 0.84. So 0.93, 0.84. And the Affirm AHF trial had a hazard ratio of 0.80. Confidence interval, 0.98. They also report heart failure hospitalizations for two of them, 0.80 and 0.74. The other primary endpoint in the FAIR HF2 trial. So walking into this trial, we've got 3 other trials that demonstrate 0.93, 0.84, 0.80 for hazard ratio for that primary endpoint. All positive, one of them significant, one of them borderline significant, another one 1.06 for the upper bound of this. And now this trial, the confidence interval for that endpoint is 0.79 with an upper bound of 0.99. That information altogether, this treatment, as the read of a statistician, this treatment works. We are sitting there with 4 trials all analyzed separately. There is a meta-analysis published. Does it move clinicians? So I don't know the answer to that. Other people can tell me that. But if I randomly pick up this article and I read it, it says this treatment doesn't work. Boy, and I know, and JAMA is an incredible journal. I know multiple editors for JAMA. They do an incredible job with it. And I know exactly how we got here. And this is not unique. We see this commonly. I struggle with it. I don't think the conclusion is right. I think it's actually highly likely the conclusion is wrong. Do people read the conclusions? So that's my struggle. So what could the world be different? Okay, Scott, so what would be different here? What can we propose different ways to do this? Well, let's suppose we didn't think of the trial as being this black and white, where we do a significance test. If it's significant, we all wave flags and we all celebrate and we publish it and it changes clinical practice. If it's not significant, it doesn't change clinical practice. Suppose what would be different? What if the trial reported the posterior probability the treatment is superior to the control? And it doesn't put an adjective on it. It doesn't say significant, borderline significant, highly significant, 3 asterisks on it. We don't have to do that. 98% is an adjective. And it allows somebody to consume the data if they want to only look at that trial. Okay, 98% probability for the primary endpoint of superiority. By the way, that satisfies the likelihood principle in that scenario. And we're overly stuck on type 1 error that it's significant. What if that's the report? And the little cartoon in the front of JAMA says, this trial demonstrated a 98% probability that it benefits time to first cardiovascular event and heart failure hospitalization. Now, the first thing a frequentist is going to say, well, oh yeah, but now you've got a prior for that. An important part of this, the important part is where this sits in the science. And I think the article in the Journal of the American Medical Association shouldn't just report on that single trial. The trial should prospectively define a relatively non-informative prior so that the data speaks for itself. That we know how to do that. It's common that we do that. And here that would give a 98% posterior probability. The trial prospectively defines a pessimistic prior. It prospectively defines an optimistic prior. This would be relatively easy to do. It also specifies a prior based on the current summary of scientific information. It uses the meta-analysis that was published. And it says, based on the previous data, and it might even have several of these, based only on trial 1, here's what you do, other trial 2. So if you don't like trial 3 and you like trial 1, here's several priors. And somebody that uses those priors, here's the probability. And my guess in this scenario is that this is 99.9+% probability this treatment is beneficial. If you use that summary of that information, updated, which Bayesians do based on this trial, and allows the reader to judge it on this, and never says it statistically significantly affects the clinical outcome, but says there's a 99.3% probability this benefits, or this trial demonstrated a 98% probability that it benefits. A pessimistic prior would be 94%. An optimistic prior is 99.3. I'm making these numbers up, but I'm just guessing what they might be. And the prior based on a summary of the other trials and this one together, that's where I think this would be 99.97% probability of benefit, something like that. As a statistician, I'd be much more comfortable when I read this article that that's providing this advice to clinicians. Where right now, when I read that article, I really struggle. And boy, I hope they look at the data. And I hope that they consume all of this information. It's hard in the setting. So this is my read of a random article that sort of stuck with me of the results of this. And it stuck with me largely as somebody who's spent 25 years in clinical trials doing publicly funded trials, privately funded sponsor trials, NIH-funded trials, patient organization-funded trials, comparative effectiveness trials, that I struggle with the scientific outcome of this. So we are not in the interim here. We are at the end of the trial thinking of it. Maybe this could have been in the interim and the trial could have been adaptive in the world we live in, but also thinks about the world we live in. Could we do things differently as we're moving forward in this and we get more and more results? Could this look different? So I am Scott Berry in the interim, and until the next interim, thanks.